Hello students, in this PowerPoint presentation we will discuss the study of anti-ulcer activity of a drug using Pylorus ligand rat model and NSAID induced ulcer model. This experiment is given in the B Pharmacy 6th semester. So in detail we will discuss the requirement for this experiment will require like albino rats 200 gram of Igersex drugs like given in the PPT that we will use Pylorus ligand ether, anesthesia, ranitidine, and for NSAID induced ulcer we will use aspirin or diclofenac, whatever you want to use. The specific dose of that drug you can use in this experiment, and the reagent we will use here the 0.1 normal NaOH. So student must know how you will prepare 0.1 normal NaOH solution, phenolphthalein indicator, and that offers reagent. And the instrument we will use the dissecting microscope with 10x magnification lens, burette, pH meter, and surgical instrument. So these will be the requirement for this experiment. As we know, the peptic ulcer is one of the most prevalent gastrointestinal disorder, and the aim of the present study is to demonstrate the anti-ulcer activity of drugs using Pylorus ligand or SHAY rat model. Why we called it SHAY rat model? Because a scientist named SHAY who firstly worked on this Pylorus ligand model. That's why due to the name of that scientist we sometimes say this model to SHAY model, SHAY rat model. So we can say this was first demonstrated by SHAY in 1945. Ligation of rat pylorus result in gastric acid accumulation. Means what is the mechanism behind why we use the Pylorus ligand? So student must understand if we ligate the pylorus of rat stomach, the gastric content accumulation in the forestomach leads to acute gastric ulcer. और ये जो mechanism होती है, this is responsible for causing ulcer. This procedure is used to screen the drugs for the anti-secretory and anti-ulcer activity. So as we know, we have different type of ulcer: gastric ulcer, esophageal ulcer, and duodenal ulcer. Why we call this is SHAY model, I already discussed. So this image is representing, this is the this is the site where we ligate का means thread से हम इसको knot लगा देंगे यहाँ पे. So the gastric content due to the effect of stress or due to the gastric content which will not be moved further, which will accumulate in this area, will cause the ulcer in this wall. So this is the normal mechanism, and same thing I will discuss for the Pylorus ligand का. आपको clear है Pylorus ligand से अल्सर किस तरीके का हो रहा है. NSAID induced ulcer कैसे कर रहे हैं? Because all the NSAID basically we know as NSAID perform their mechanism by inhibiting the COX enzyme, and COX is responsible for synthesis of prostaglandin. Especially when we talk about the ulcer, the prostaglandin PGE2 is responsible for making the mucus layer. So if we use the NSAID, the PGE2 synthesis will be inhibited because the COX will be inhibited. So there will be no formation of protecting mucus layer, and that will lead to the gastric ulceration. So this is the mechanism of NSAID induced model. So what will be the procedure? First we will talk about the Pylorus ligand SHAY rat model, and then after we will discuss NSAID induced model. So here we are discussing the Pylorus ligand model. So in this procedure we will take albino star rat, n is equal to 6. We will divide them into two groups, and we will keep in mind that animal must be fasted with free access of water. What we will do in the second day we will ligate. सेकेंड डे जब हम उसको लाइगेट करेंगे तो उसके पहले मीन्स अभी हम लाइगेशन की कोई बात नहीं कर रहे हैं. एक दिन हमने इसको 24 आवर्स के लिए फास्टेड रखा है. दूसरे दिन हम उसको क्या देंगे? कंट्रोल. दो ग्रुप होंगे हमारा. ग्रुप फर्स्ट को हम कंट्रोल ग्रुप बोलेंगे और ग्रुप सेकंड को हम बोलेंगे स्टैंडर्ड ग्रुप. तो स्टैंडर्ड ग्रुप में हम रैनिटिडिन देंगे और ग्रुप फर्स्ट में हम कंट्रोल ग्रुप में सैलाइन देंगे. तो ये हमने दे दिया, और उसके जस्ट देने के 30 मिनट बाद मीन्स आफ्टर 30 मिनट हम क्या करेंगे? रेनिटिडिन एक्सपोज करने के 30 मिनट के बाद मिड लाइन इंसिजन करेंगे और पाइलोरस को उस पर्टिकुलर जगह पे लाइगेट कर देंगे, राइट। अब पाइलोरस को लाइगेट करने के बाद हम उसको फिर से स्टिच कर देंगे और स्टिच करने के बाद रैट को हम इंडिविजुअली विदाउट फूड एंड वाटर उसको हम इंडिविजुअल केज में रख देंगे, सारे रैट को इंडिविजुअली अलग अलग। और रखने के 4 घंटे के बाद आफ्टर 4 आवर हम एनिमल को डीकैपिटेट कर देंगे, ओपन करेंगे स्टमक को फ्रॉम द ग्रेटर कर्वेचर, कलेक्ट द स्मॉल कंटेंट इन द सेंट्रीफ्यूगेशन ट्यूब एंड क्लीन विद सैलाइन। तो हम जो स्टमक होगा उसको हम नॉर्मल सैलाइन से क्लीन भी करेंगे। And then only after we will, by using the different mechanism, we will determine the ulcer index, ulcer score, total acidity, free acidity, and under 10x magnification, and ulcer to be observed. So this is the normal procedure for the Pylorus ligand model. How we will calculate the ulcer index? By calculating the ulcer index, there is a formula that u1 plus u2 plus u3 into 10 to the power minus 1, where u1 indicate average number of ulcer per animal, u2 is equal to average of severity score, and u3 is equal to percentage of animal with ulcer. So here the scoring of ulcer will be done on the intensity. Like if the normal coloration is there, we will score 0. For red coloration, this is 0.5. So this is the various point you can note it from here. And the ulcer score, if it is of 1 mm exactly, then we will give the score 1. If it is lie between the 1 to 2, we will score them 2. If it is more than 2, we will 3, and more than 3 is equal to 4. So the ulcer score will be maximum of 4. In the second, how we will calculate the side by side, we also calculate the pH through the pH meter. Analysis of stomach content. So for the analysis of stomach content, first we will measure total volume of gastric content and centrifuge at 1000 rpm for 10 minutes. Pipette out 1 ml of supernatant liquid of the centrifuge content and dilute with 10 ml of distilled water. Titrate the liquid against 0.01 normal NaOH using Topfer's reagent as indicator till the end point. End point means appearance of orange color is obtained. The volume of NaOH used to be noted to estimate free acidity. Titration has to be continued till the appearance of pink color, and the volume of NaOH run down is to be noted to calculate total acidity. So this is the simple flow chart where you can easily understand how you will calculate the free acidity and total acidity. And after this analysis of stomach content, or you can say the total acidity and free acidity, you will draw a table like observation table. Here you will mention the control group and the standard group, which you have given the ranitidine 20 mg per kg per oral. And the volume of gastric content you will use, simply you will measure from the whatever amount of the pH you obtained in both group, free acidity, total acidity, and the ulcer index. And the conclusion will be right that let's suppose you have you will make your conclusion from here, like the volume of gastric content, pH, free acidity, and total acidity and ulcer index for control and for standard. So both you will compare, right? After the comparison of both group from each other, you will reveal your result. So this is the conclusion, and here are the various references that you can write. That I have written the content from these sources. So first paper I have added here from the SHAY. So here you can use these references for your purpose. Question you should know in the last of this experiment, like how does the student will clean the stomach, how we measure the size of ulcer, define free acidity and total acidity, how does NSAID induces ulcer, how Pylorus ligation induces ulcer. So these are the mechanism. I hope कि आप लोगों को ये आने लगा होगा. Now the procedure for the NSAID induced ulcer, very simple procedure. In the same way we will take 6 rat and divide into 2 group. Group first receive the 1% of CMC, carboxymethyl cellulose, and while the second group received ranitidine 30 mg per kg per oral. After 30 minutes of drug administration in both group, administer aspirin or diclofenac, whatever you want to take. If you will give aspirin, this will be 200 mg per kg, and diclofenac it is 100 mg per kg, suspended in 3 ml of CMC. So as we are suspended these drug in the CMC, that's why we are giving CMC in the group first, control group. क्योंकि हम कंट्रोल ग्रुप में वही अगर हम कंट्रोल ग्रुप में नॉर्मल सलाइन देते तो हमारा बट ऑब्वियस था कि रेनिटिडिन को जो हमने डिसॉल्व किया या जो हमने एस्प्रिन या डाइक्लोफेनाक को डिसॉल्व किया वो सलाइन में डिसॉल्व किया। क्योंकि एस्प्रिन और डाइक्लोफेनाक को हम एस्प्रिन या डाइक्लोफेनाक जो भी हम लेते हैं उसको हम सीएमसी में डिसॉल्व करेंगे, इसलिए हम ग्रुप फर्स्ट में सीएमसी दे रहे हैं। अवॉइड फीड एंड वाटर आफ्टर 6 hours, सैक्रिफाइस द animal through cervical decapitation. Open the stomach and wash from saline. Calculate the ulcer index. Because we have already calculated the pH, volume, gastric content, free acidity, total acidity, and ulcer index. So in this NSAID induced ulcer, we will calculate only ulcer index. I have given all the detailed information that how we will calculate the ulcer index in a previous slide. Here we will prepare the observation table mentioning that only the ulcer index. So the result we will calculate the obtained data in this NSAID induced ulcer or in the Pylorus ligand model. Obtained data reveals that comparison of ulcer index, ulcer score, pH, free acidity, total acidity concludes anti-ulcer effect of ranitidine in both models. So this is all about the Pylorus ligand. This is all about the study of anti-ulcer activity of a drug using Pylorus ligand and NSAID induced rat model. Thank you very much.